name is Marcus Teat. I'm a NOAA CSC student at Delaware State University. You'll be fooled by the name tag. It says NOAA, but actually it's not CSC. And the title of my research is Eastern Oyster Restoration, Recruitment, and Their Impact on Species Diversity, Water Quality, and at Rehoboth Bay, Delaware. So before we can get into water quality and like how we did species diversity in Rehoboth Bay, we have to give a background information on our site as well as our Eastern oysters, or Pascharia virginica. They are native to Eastern North and South America, as well as parts of the Gulf of Mexico. They're able to provide multiple keystone, they're keystone species that provide multiple benefits, such as they are able to provide habitat and food for juvenile fish crustacean. They improve species abundance and diversity. They are able to improve water quality by being filter feeders. They also protect coastal habitats or reduce erosions through the use of reefs. And they are also, we also love to eat oysters. I don't know if anyone likes to eat oysters, but they're good for the economy as well. Unfortunately, over the last couple of decades, oyster populations have been on the decline. This has been due to two separate reasons. One of them has been through parasitic diseases. The first one being in the 1950s due to MSX. You can see on the graph right there where exactly mortality for MSX began. And as oyster populations started to decrease, another parasitic disease called dermo, parasitic disease called dermo in the 1980s happened, which caused the oyster population to decrease even more. Another reason that oyster populations have been on decline has been through anthropogenic factors, through overharvesting, habitat destruction, and water quality degradation, the increase of nitrogen and phosphorus within the water, resulting in a decrease in dissolved oxygen and killing off oysters. So we did our research in Rehoboth Bay, Delaware. It's part of the Delaware Inland Bay System, along with Indian River Bay and Little Assawan Bay. It is connected to the Indian River Bay to the south and the Rehoboth Canal to the north. It has multiple shoreline features such as agriculture, boathouses, commercial marshes, and barrier structures that negate wave energy. And it's also a highly populated area during the summer months. We don't have a lot of people here, but show of hands, who likes to go out in the water? Who likes to go to the beach during the summer? Yeah, basically everyone. Yeah, it's summer months. Everyone wants to go out. Everyone wants to go to the beach. Everyone wants to go out boating, fishing, whatever. Next to Rehoboth Bay, we have Rehoboth Beach, which is a very popular place to stay during the summer. So between May and June, July, August, this gets very heavily populated, and there's a lot of traffic. There's a lot of boats out in the water or out in the ocean and whatnot. There has been restoration efforts in Rehoboth Bay. I know Anthony, the talk before, was talking about in Texas how aquaculture is a new commodity there. Well, that's not really that much different from Delaware and Rehoboth Bay itself. Only a couple of years ago, aquaculture has really began. And there has been aquaculture by companies such as Rehoboth Bay Oyster Company, who harvest oysters to sell. There's also has been restoration efforts by a nonprofit organization that we collaborate with. They are called the Delaware Center for Inland Bays, or CIB for short. And they have deployed multiple pilot artificial oyster reefs in the Delaware Inland Bays. They deployed 2 in Rehoboth Bay that I will talk about later. And I will talk about how we monitor species diversity oyster recruitment. So the objectives of this research was to monitor and compare the effects of artificial oyster reefs and aquaculture sites have on biodiversity and oyster recruitment, and as well as to observe physical and chemical water quality parameters and how oyster reefs and farms affect them. And one of the research questions is how effective are CIB's pilot reefs for improving water quality, species diversity, and oyster recruitment in Rehoboth Bay, Delaware compared to natural sites, as well as oyster farms. So going back to Rehoboth Bay, this is our study area. We have 7 different total sites that we split off into 3 different groups. We also color coded for the sake of clarity. We color coded the groups as well. For Camp Arrowhead, Big Bacon Reef, also called Big Bacon Island, there are artificial reefs. They are usually labeled in blue, or they will be labeled in blue for this presentation. And you can see they're right there on the west side and east side of the bay. We also had two aquaculture sites called Sally's Cove and Rehoboth Bay Oyster Company. They're labeled in green. We also have two control sites in Bay City and Sally's Cove Control that are going to be labeled in yellow and red. And we also have one extra site called Rutherford Control, which is actually a former aquaculture site. It used to be owned by a farmer called Chris Rutherford, but ever since then he has actually left the site. So now we have basically a site that is not necessarily a true control site, but it's also not a true aquaculture site. So in terms of how we monitor biodiversity and recruitment, we took oyster shell bags from a CIB-owned shell pile, and we deployed 4 bags at each site. And these bags were left in the water for about 5 weeks before they were retrieved. And then we took them back to our lab, and then we sieved them to monitor oyster recruitment. We monitored what species we found, and then we also monitored species abundance as well. We also did water quality testing both on site and in the lab as well. For the sake of time, I'm not going to talk about every single water quality permit that we have done in the lab, but I will talk about the ones that I will be talking about in the presentation. We have done water quality with a YSI 5x6 multi-probe handheld that tests multiple water quality parameters such as ORP, pH, conductivity, salinity, and temperature. We also done testing with a Turner Design aquapharometer and turbidimeter to test chlorophyll A and turbidity. And we also do analysis in the lab for chemical water quality parameters such as phosphate, nitrate, ammonia, calcium, etc. For the sake of time, I'm only going to be talking about dissolved oxygen, orthophosphates, and nitrate. So here are some of the results that we've had. This is for oyster recruitment, and through the summer months of 2021, we found that oyster recruitment was the highest at the artificial reefs, and then we also found that it was the lowest at our former aquaculture site, Rutherford Control. And then on the graph on the left, you can see how each individual site fared. And our lowest site was at Bay City, and that performed the poorest, which wasn't very surprising to us given where Bay City is. It has a lot of boat traffic. It's right next to a pier. It's very difficult for any oyster recruitment to basically happen there. And then we also saw our 2 best performing sites were our artificial reefs on the left. You can see that it was Big Bacon Reef and Camp Arrowhead. For species abundance, we once again saw that some of our artificial reefs performed the best, particularly Camp Arrowhead. We once again found that Bay City again performed the poorest. And on the next slide, we have some of what the dominant species were, including mud crabs, barnacles, grass shrimp, and Atlantic oyster shrimps. We also monitored species richness as well, and we found that it was again the highest at the artificial reefs. It was then second highest at the aquaculture sites, and then lowest at the former oyster farm. And for species diversity, we found that in our control sites overall performed well with species diversity, but then you look on the left side of the graph, and you look at some of the best performing sites individually. Sally's Cove Control was one of the sites that performed the best alongside with Sally's Cove and Big Bacon Reef. And Bay City performed adequately, but once again, Rutherford Control is one of our poorest performing sites. Onto water quality, and we found that in terms of comparing between each site, We found that Bay City was at times one of the highest performing sites in terms of dissolved oxygen. And then we also found that if you look at some of the other sites or some of the other parameters for nitrate and orthophosphates, some of our artificial reefs actually actually had some of the highest nitrate levels, especially during, say, the October months, where Big Bacon Reef and Rutherford Control was what had the highest nitrate. And onto orthophosphates. Once again, we saw our reefs were at times all over the place. We had times where Bay City, one of our control sites before, had the lowest amount of orthophosphates. Sometimes they had the highest. At times it was our BOC, Rehoboth Bay Oyster Company, or Sally's Cove, our aquaculture sites. And then at times, like especially during November, it was the lowest again. So in summary, overall oyster recruitment was the highest at the artificial reefs, followed by the aquaculture sites with Camp Arrowhead and having the highest recruitment. We also saw species richness was the highest at the artificial reefs, and the second highest at the aquaculture sites again. However, species diversity was the highest at Sally's Cove and Sally's Cove Control, followed by Big Bacon Island, our artificial site. And then overall, the aquaculture sites for species diversity performed the best, followed by the control sites. While the former oyster farm performed the poorest. And then Bay City showed the lowest nitrate levels during June and August, while the high nitrate levels at Big Bacon Reef and Rutherford were in October. And the lowest levels of orthophosphates and artificial reefs for June and September months. So in conclusion, restoration efforts through oyster reconstruction has increased wild oyster recruitment at Big Bacon Reef and Camp Arrowhead sites. However, and as well as the presence of structured habitats has shown increased species abundance and diversity across the bay where we had artificial reefs and oyster farms. However, we're still assessing the relationship between water quality and oyster or no oyster sites. However, we believe that given time, artificial reefs as well as agricultural sites should be able to help water quality through filter feeding and nitrogen storage. I would like to acknowledge my program director, Dr. Stacy Smith. I also like to acknowledge my advisor, Dr. Billy Osbe, and as well as mentorship provided by Memory Nakasway and Aaron Bland. I would also like to thank my research partners, some of them are in the room right now. Thank you. And I also like to say special thanks to Zach Moore and Andrew McGillman from the Delaware Center for Inland Bays, as well as Grant Blank at Delaware State University for help providing us with transportation and overall help with our sites. And I would also like to say thank you to NOAA and Lamar CSC. And with that, I would happily take any questions.
Over time for a couple of questions. Any questions?
Marco, it's a great talk. So you look at, excuse me, you look at water quality influence in recruitment of oysters, but given that these are filter feeders, I would assume that water exchange flow may also affect the recruitment. How would you measure, if you had to measure flows in the different areas in order to get an idea whether the amount of water going over a place will influence the recruitment of oysters?
So you're asking me I'm sorry, repeat the question?
Yes, how given that these are filter feeders,
right.
Obviously water quality must be one really important parameter, but the other one I would assume how much water exchange is there.
Mm-hmm.
They tend to place the oyster farms in places where there's really good water exchange so that they can filter lots of water and get a lot of plankton.
Mm-hmm.
How would you measure water exchange, or what kind of data would you need to get an idea on different current tidal exchange, water exchange in each of the locations?
That's a very good question, and that's a question that I actually don't know the answer to. I know for our future research, we're planning on working with oyster larvae dynamics, wondering how oyster larvae enters in and out of Rehoboth Bay. But that's not something that we have actually yet discussed yet, but I would definitely talk to Dr. Osby.
We also, the high tide and low tide, we try to do the consistent monitoring at certain tides. And in his case, mostly closer to low tide. Because there is a big variation in water quality parameters.
Yeah.
And
I noticed that one of the measurements, the parameters, was poorest at the, I guess, abandoned oyster aquaculture site. Do you have any thoughts about that? Like whether it was suitable or what does it?
Well, that site, some of the sites are slightly different in terms of like how much boat traffic goes through or how much they're affected by other coastal like areas. For example, like I was talking about Bay City, and Bay City is like one of our most heavily populated sites. My assumption would be that at least like that site specifically still gets a lot of boat traffic back and forth. That might have some contributions to why water quality at times there is very poor, even though it is a former aquaculture site.
Okay. I was just wondering if the oyster farmer left it because it wasn't a good site.
Probably.
yeah, it might be. The other thing is that he didn't leave anything. There is no structure except 2 poles. Like this is the beginning and ending up in the water. That's
Not
all.
even those poles are gone now.
They removed that too?
Yeah,
Oh,
I remember they were there. We went out to that site a couple of times. We were using the pole for references. And then one time Aaron, our former lab technician, drove the boat to that site, and then he was looking around, looking for the poles. It's like, where are they?
All right, thanks.
Any more questions? Thank you.